I am Suzanne Godby-Inglesby, the Associate Director of IU's Institute for Advanced Study, and it is my pleasure to welcome you to this morning's webinar on identifying changes in the Wabash River corridor's vascular plant flora, which will be presented by Rich Hull. Before we begin today, I would like to note that the IU Institute for Advanced Study recognizes that Indiana University is built on the ancestral homelands and of the Miami, Delaware, Potawatomi, and Shawnee people. We acknowledge and honor these indigenous communities, both past and present. To learn more about increasing representation of and support for Native and indigenous communities, I encourage you to look into Indigenous Indiana and to also explore the resources available through IU's First Nations Educational and Cultural Center. Today's format will involve a presentation followed by a question and answer period. You will have the opportunity to put your questions and answers or questions and comments in the Q&A link that hopefully you see at the bottom of your screen if it's a full screen, and I will share those with our presenter. Typically, we hold those until the end of the presentation, but if you have something that is about a specific slide or point that was just made, if you will go ahead and put that in there, I will try to insert that so that we can get it answered in real time, but otherwise we will come around to those at the end of the program. This program will be recorded and available for later access so that you can come back to it. That will be posted on our website, which we encourage you to visit. We're IAS at Indiana University, so ias.indiana.edu. Our Resilience in Memory in Archives, Libraries, and Museums series will continue next semester. So for more information about that and to register for those webinars as well, please look on our website for that information too. We will share the link in the chat in just a little bit, so you'll be able to see that. And now it is my pleasure to introduce today's presenter. Rich Hull is a second-year graduate student in the Knox Lab here at the Department of Biology at Indiana University. Rich is originally from Northern Georgia, where he received his Bachelor of Science in Ecology from the University of Georgia in 2020. Botany is Rich's primary field of interest, and today he'll be talking about his dissertation research on the flora of the Wabash River in Indiana. Specifically, this talk will focus on the project's overall importance, the preliminary field results from 2021, and the future goals of the study. So Rich, thank you very much for being with us. We are looking forward to your presentation. I'll turn it over to you now.
Thank you very much for that introduction. I'm going to go ahead and share my screen. There we go. And then, does that look good?
Yep, you're good.
Great, thank you. So thank you for the introduction. As stated, my name is Rich Hull. I'm a member of the Knox Lab here in the Department of Biology at Indiana University. And today I'll be talking about my research identifying changes in the Wabash River corridor's vascular plant flora. As the title would suggest, I focus my research on the western Indiana Wabash River. And right now I'm constructing a modern flora of the region to update the status of rare and exotic species, to perform a comparative analysis between the historic and current floras, and then to use this information to predict the flora of the future at different time intervals. So here on the right is a figure of the study area, the 10 counties where I have field sites, and then later on I'll go into more detail about that. So taking a step back, today I will be specifically addressing the intellectual merit for the project. So I'll be talking about the importance of vascular plants and the impact anthropogenic forces is having on them. I'll be talking about why I'm doing this research specifically in Indiana. I'll be talking about the contribution of the Indiana University Herbarium. And I'll be talking about the methods for the current vascular plant collection. This will be followed by the preliminary results from the 2021 field season, as well as future goals, which will specifically focus on data analysis once the entire collection is finished. And here on the right, I have a picture of a Simina triloba, the Indiana banana, and I'll sprinkle in a few photos throughout the presentation. So first, the basis for this research is broadly that vascular plants are vitally important. There's about 500,000 species of vascular plants worldwide, many of which are primary producers in terrestrial ecosystems. They convert CO2 to sugars, convert water to oxygen, vitally important ecosystem services, and they also contribute to most of the biomass on Earth. So here on the bottom left, I have a figure showing that plants comprise about 450 gigatons of carbon, compared to only 2 gigatons of carbon comprised of by animals. So in addition to being very important for ecosystems around the world, they are under stress. There are three main points of stress I think of traditionally. So the first is habitat destruction and fragmentation. The second is anthropogenic climate change, where the climate is changing faster than it has in historical record. And then invasive and exotic species. And this last one is known to be the most influential, at least now, anthropogenic force. To illustrate this, I created a bar chart of the Indiana flora divided into native species and non-native species. So here on the right, we have the native species, and here on the left, we have non-native species. And you'll notice that the non-native species are abundant. We have about 800 exotic species here in Indiana, comprising about 30% of the entire state's flora. So overall, there is a need for determining how native floras are responding to each of these anthropogenic forces. But despite this, most plant research is focused more on systematics, population genetics, evolutionary biology. Recently, as advancements in this field has made it very popular and more effective. And some of this research does touch on anthropogenic forces and is very beneficial. One other aspect of this is modeling, especially species distribution models. But overall, these lack large-scale empirical studies focusing on biodiversity. These are really becoming less common in academia. So that's where my research comes in. It's a large empirical study that provides a framework for analyzing how the native flora of the Western Wabash River corridor here in Indiana is changing. So then the next question really is, why did I select Indiana? Why the Wabash River? And there are a number of answers to that. The first is that there's a large climate gradient over this region. So I can analyze to see whether plants are moving latitudinally in response to climate change. There's similarly a physiographic region gradient, where we can see if plants are able to move out of ecoregions that they may be well adapted to into regions where they may not be as well adapted to. We have patchwork habitat, so we have land cover that is not necessarily conducive to plant migration, but we want to see if they're able to migrate anyway. The same thing goes for a large amount of human activity. We want to see the degree of resilience that the native flora has to agriculture and other human impacts. And then lastly, and maybe most importantly, we have historical records that enable a comparative analysis between the past flora of the first half of the 1900s to now. And this is vitally important. So here's the Indiana study area blown up. The purple counties are where I have field sites. You'll notice that the Wabash River here flows in from Ohio to the west, and then starts to go southwesterly. And I pick it up in Tippecanoe County and follow it all the way down to Posey County. And this is a latitudinal gradient of about 200 miles. And this is important because it allows me to have climate gradient or a climate gradient. So here I have mapped on the right historical minimum average January temperatures for the time frame that the historical data was collected from 1896 to 1952. And you'll notice that there's 5 different segments of 1 degree Celsius intervals, starting with -4 to -5 degrees Celsius here in the southern area, and -8 to -9 degrees Celsius in the northern area. And this is fascinating because most of the variation in minimum January temperature in the entire state was present in this area, despite it only being 10 of the 92 counties here in the entire state. So there's only one more colder latitudinal segment in the state, and we have 5 out of 6. So similarly, with ecoregions, we can see if a plant will move from the interior river, valley, and hills here in green up into the eastern Corn Belt plains, or vice versa. And we can also look at finer variation at the ecoregion level in level 4 ecoregions. So here on the right, I have land cover. And as I alluded to earlier, the area has been highly impacted by agriculture primarily. There's some development in Vigo and Tippecanoe counties as well. But here in the southern part of the area, we have natural areas that are essentially islands. They're isolated, they're typically smaller. We want to see if plants are migrating despite this in the area. In contrast, in the northern part of our study area, we have more contiguous riparian corridors that could allow for easier plant migration. So it'd be an interesting kind of comparison within the study area to see if we see differences there. This ties right into the impact of humans on the area. So here's a picture of the Wabash Erie Canal, which ran along the Wabash River during the late 1800s. And this was actually a financial disaster eventually, but also was not good ecologically and introduced many invasive aquatic species to the area and such. So when we think about human impacts that could demonstrate the degree of resilience the native flora has, we think about water diversion, transportation, agriculture, those sorts of things. So lastly, the really important component of this research is having historical data. So the herbarium here at Indiana is a research collection where voucher specimens are stored. And these voucher specimens are physical preserved specimens of plants that literally vouch that a plant existed at a specific location at a specific date. And the herbarium here holds 155,000 of these. Importantly, these are digitized specimens. So they're images online of these, and the label data is transcribed and it's available for research. And this was a large effort done by Dr. Knox here at the herbarium and Dr. Paul Rothrock. 87,000 of these vouchers are from Indiana. And then 48,000 were collected by Charles and Stella Deem from 1896 to 1952. So I'll take a break here and briefly introduce Charles and Stella Deem. Charles Deem is a state forester of Indiana. He was actually the first state forester in Indiana. He started as an amateur and delved into botany really, and eventually decided to create a flora of Indiana where he would have county-wide distribution data for each species. So he started this in the early 1900s and published that in 1940 and still collected until his death in 1952. And Stella Deem would collect with him, collect sometimes on her own as well. She was his wife. So collectively, they both produced 6,100 vouchers from the study area. So this really provides a window into the past for us to perform a comparative analysis. I wanted to illustrate that this data isn't just available to myself as a researcher. It's available publicly to the public online. So here we have the website, the Consortium of Midwest Herbaria. So this website contains digitized data from herbaria throughout the Midwest. And if you go to this URL here, you'll have to scroll down to the Indiana University checkbox and check it. And then if you go to the next page, you can put in your search query. So I put in Indiana, Posey County, the collector's last name of Deem, and I put in the time frame, 1896 to 1952. And once you search that, it will provide a list of the specimens collected then. So here we have just over 1,200 specimens actually collected in that fit that search query. So here we have up top Botrytis virginianus. And you'll notice there's some general data here, and there's a digitized specimen there. If you click on that image, it will bring up the complete digitized specimen. So here we have the preserved plant, and then importantly, we have label data here. So this is the old name, Botrychium virginianum. It's since been changed to Botrytis virginianus. And it was collected by Charles Dean in Posey County in the very southwestern portion of the state back on May 25th, 1906. And it also includes his collection number and a general location. And this general location is very important because this data was taken by the university and, well, by the herbarium and georeference. So we now have approximate GPS coordinates for each of these specimens. So this is very important for spatial analysis. And this is not something that is standard for many herbaria across the United States. So if we go back to this page here and click on maps, that brings us to the GPS coordinates for all these specimens that fit that search. So here, the red dots represent one specimen or maybe many specimens, and they're all in Posey County. So altogether, the historical data combined with the attributes of the study region enabled me to address many research questions, which are not just limited to the ones I present in this talk, but these are some of the basic ones I want to talk about today. So we have kind of the standard floristic study questions, like has the species richness of exotic species increased in the study area over time, or have rare species declined? But we also have more sophisticated questions that are more complicated, especially statistically. So are native species moving latitudinally in correspondence with climate change, for example? We also have land fragmentation questions we want to address, as well as just some general quantitative biodiversity questions. So this leads me to hypotheses or predictions for these questions. First, I expect that exotic species will be more biodiverse than in the past because there's been increased human activity in the region, more transportation. Worldwide transportation has continued to increase over time. Secondly, rare species will be slightly less common than in the past. I don't think they'll be much rarer because there are high quality regions maintained by land trusts in the state and by the Indiana Department of Natural Resources. And then moving on to the climate question, I think that some native species will have moved due to climate over time. And these native species are likely to be species that are well adapted to disturbed areas because these areas are frequented by humans often, which transport seeds over large distances to areas that they may be well adapted to, these disturbed areas. So if you think of roadsides, it's a good example. And previously, the only thing holding them back was they just couldn't grow in colder climates, for example. So we expect some southern species to move northwards because they're well adapted to disturbed areas. So moving on to land fragmentation, we expect that both land area and the time since last disturbance within the plot will impact the composition of the native flora. We expect that several factors will impact beta diversity, and then we expect that greater phylogenetic diversity will be present in the southern and northern sections of the study area. And these last three questions, really, I'll go into more detail in the future directions and explain exactly what I'm talking about. So right now, I'll go ahead and move on to the process for constructing the modern flora of the study region. And the first thing I'll talk about here is the site selection. So for each of these sites, I'm constructing a site flora. So I want a complete list of the vascular plants at each site. And for each species, I want photographs as well as a voucher specimen that will be deposited at the Indiana University Herbarium. So in total, I have 50 sites, and these are represented by red dots here on the figure to the left. And these sites vary in many different ways. So they vary in size, ownership, etc. but they were selected because of their natural habitat. So I looked at aerial imagery. I selected plots that looked like they had good natural habitat still present and that were close to the Wabash River. And then I figured out which ones were accessible. So believe it or not, 50 sites was not a preconceived number. It was simply the number of sites that I could find that had a large amount of natural habitat that were close to the Wabash and that were accessible to me so I could perform research there. So as I mentioned, sites are diverse. They include roadsides, county parks, nature preserves, state parks, etc. And I access each site with permission to collect plants granted from the Indiana Department of Natural Resources as well as the property owners. So here I have a couple examples of sites. Here's the Miram Bluff Park in Sullivan County overlooking the Wabash River here and into Illinois. It's a very neat site. I found a couple of county records there this last year. And here I have a hemlock stand in Park County overlooking Sugar Creek. So that's a new nature preserve called Mossy Point Preserve. So now I'd like to move on to my collecting strategy for each site. So here I have a map of Twin Swamps Nature Preserve. It's a very nice preserve maintained by the Department of Natural Resources down in Posey County. And the numbers here actually represent historical collections. So I have a key that tells me what specimens were collected there, etc. But my general strategy is to look at aerial imagery and try and find the different habitats within each site. So here on the left, we have some second growth forest that's fairly visible, and that is most likely going to have a very different species composition than this more contiguous older forest here down in the bottom left or between these two swamps. And even this swamp here is different from this swamp, because this swamp, you can tell from the aerial imagery, is evergreen. So this is bald cypresses. And then here we have an overcupped oak swamp. So trees lose their leaves in the winter. So the goal is really to sample evenly between these different habitats to generate a representative list for each site. As for actual voucher collection, I judiciously collect. So I do not collect a plant if it will endanger the population, whether the plant is rare or is not rare. So in that case, I'll just take a photo. If the plant is plentiful at the site, I'll collect the whole plant if it's herbaceous. So the whole plant is potentially needed for identification. And then I collect clippings from woody plants, such as this redbud tree right here. So here are some photos taken out in the field this past August by George Armstrong, a master's student here at IU. And this is at a Portland Arch Nature Preserve. So here I am taking notes. So again, notes provide the data for these labels that I showed with the Charles Deem specimen I'll be showing on these next slides. Here I am with a nice fern, and then here's the pressing process in the back of my truck with a plant press and blotters and a lot of material to press. So again, another picture of the truck filled with a site's worth of plants here on the right, and then the plant press here on the left with cardboard and newspaper and blotters, all for properly pressing a plant. Here's a picture of some of my field notes. This is what it looks like. Each number is a collection number, so that's specific to that specific specimen, and then I'll give it some data as well. And believe it or not, these somewhat messy notes end up making good labels later on. I'll show that here in a slide or two. So plants are initially dried in blotters that are within these plant presses shown here on the right. Plants are very moist, and you have to remove that moisture in order to preserve them for a long time, or else they'll molder, basically. And then once I get back to the herbarium, I take the blotters out. I press the plants in a dryer for about 3 days. From there, they're frozen actually in a very nice freezer for 5 days. And this is done to kill any insects that may be on the plants, any insect larvae, any mold. And from there, they await mounting. So here is one of my specimens from this past year. This is actually the same plant in the field. This is a shooting star species, the one we have in the eastern United States. Well, one of the ones we have in the eastern United States, the most common one. And then, believe it or not, the really indicative identification feature is how long the base of the leaf is, so the petiole here. And then here, the specimen's preserved on the right. It's mounted on archival paper here at Indiana University. This fragment folder is full of fragments from the plant that can be sequenced if need be at a later date. And then we have the barcode down here for just tracking the specimen within the collection. And then we have my label data. If we zoom in on my label data, I wanted to illustrate some of the information I'm keeping on each specimen. So obviously, I say I collect it. I have that specific collection number again. I have the date collected, and then I have my determination right here. I have the broad locality, so Sullivan County. I have a more specific locality, Mirren Bluff Park. And we saw this in the picture earlier. And it's located, the specimen is located along the Mirren Bluff Boardwalk Nature Trail, approximately 35 meters east of the intersection between Poplar Street and an unnamed road. And this is reflected also in the GPS coordinates I take using a GPS tracker while I'm at the site here. I always note the elevation. I note the habitat. So in this case, it's growing on wooded, steep sandstone bluffs. And then I always note the associated plant community. So I have several species that those coexisting with. It's important to write down the flower color of the specimen because this can change during the drying process. And then I also like to see or record how common the plant is at the site. So in this case, it's uncommon. Here's another example of a voucher specimen I collected. And again, this is the same plant here on the left in photographs. This is Ranunculus micranthus. And the reason I'm showing this is I want to illustrate the importance of taking voucher specimens over just taking photographs or just putting a plant on a list in research. And the reason why is I initially misidentified this plant. So I had it at Ranunculus abortivus, which only one of the main differences, probably the most visible differences, is just that the lower part of the stem here has long villose hairs. So these are basically just long white hairs that grow on the plant and fall off as the plant matures. But Ranunculus micranthus has this feature, Ranunculus abortivus doesn't, and I overlooked it. So the herbarium actually, someone at the herbarium, when they're mounting the plant, said, hey, this isn't it. So these voucher specimens really enable us to go back and change the determinations to something else, whereas this probably would have never been caught if it had been a photograph or simply a name on a list. So it's pretty neat. And then here, again, is some of my voucher data. So this is what I do for every specimen. And I wanted to show that if you go through the same process that I did for the Dean data, so if you go to the Midwest Consortium of Herbarium, click Indiana University, and then search my last name, all my specimens will come up. And although most are not mounted yet and therefore not digitized yet, I do have the label data online. I also have a lot of photographs already online as well that are of actually the same plant that will be in the herbarium specimen. I keep track of these specimens in checklists that are not available to the public. Whether or not they will be available to the public will be up to the landowners. But this really just helps me keep track of the species at each site as I go along. So I add these, and I also I can add a hyperlink to the exact specimen that represents that species, so I don't duplicate the specimen in upcoming years or on subsequent collection trips. So overall, this is the first year, this is the first collection year of 3 collection years. And I collected just over 2,500 voucher specimens that represent 107 vascular plant families, 313 genera, and then 531 vascular plant species. This represents about 20% of the entire state flora in just 10 counties. And then specifically looking at exotic species, I collected 101 species, including 22 invasive species. I collected 17 rare taxa as defined by the Indiana Department of Natural Resources, and I observed 2 more species that were too rare to collect. And overall, I collected 152 county records, which were determined to be county records after consulting publications, different herbaria in the region, and the USDA plant database. So diving into these results a little bit more, again, we had 101 exotic species, including 22 invasive species. And these were not necessarily as prevalent when Dean was collecting. So Lamium purpureum was probably just starting to show up in the study area when Dean was collecting. Taraxacum officinale, the really common dandelion, may or may not have been here. We're not really sure. And then we have Ranunculus parviflorus, which definitely was not here when Dean was here. In fact, it's only started to show up in the state in the last 10 to 20 years. This is from Posey County, actually, from a state park down in Posey County. So one of the main questions I had at the beginning of the talk was, has the species richness of exotic species increased in the study area? So I did some preliminary things just to see, if that's the case. So here, Dean collected 6,100 voucher specimens total. And out of these, only 336 represented exotic species. This is about 6%. In direct comparison, I collected 2,500 vouchers, but 598 represented exotic species. So it's about 24%. 126 exotic species were documented by Dean, including 10 invasive species. And with only 2,500 vouchers, I collected 101 exotic species, including 22 invasive species. So given Dean's number, a rough extrapolation would have been, I would have probably been expected to collect 50 exotic species total. Now, I don't put too much emphasis on these results yet because we don't have the complete data set. And for this first year, I was really trying to knock out a lot of very common species at each site. So I feel like these percentages definitely are overemphasized. So it'll be really interesting in 2 more years to see how this has changed. So these are a lot of photos. This is probably surprising. Most presentations are not supposed to have more than probably 1 or 2 photos per slide. But I wanted to illustrate what it's like to be a botanist out in nature. So here I have all 17 rare plant taxa that I took voucher specimens of for. And it's hard to find these things as you're looking through a forest, and some stand out like Silene regia, some stand out like Spagelia marilandica or Heterotheca camporum, but others like Charax socialis just look, diminutive. You would never have thought, okay, this is going to be a rare plant, so a lot of it is experience that enables you to pick these things out. A lot of it is just, you pick them up as you're going around. And again, this is why it's important for each species to judiciously collect, because you don't know exactly what's a rare plant all the time. But some of these have really neat stories too. So this picture here is of Nothoschordum bivalve. And this population is down in Posey County. And Dean collected it in the early 1900s. And I'm not sure that it's been collected. Well, I know it's not been collected since then. I'm not sure if the Department of Natural Resources knows the population is still there or not. Now they do. But it was collected in flatwoods forest. Since then, the site has been logged and it's no longer a flatwood forest, but it's still there on a roadside. So it's a remnant population. It's very neat. So another question I introduced earlier was, have rare species declined? And doing a similar thing that we did with the exotic and invasive species, Deem had about 5% of his 6,100 vouchers represent rare species. I, in comparison, had about 1%. But again, this is my first year doing field research in Indiana, and finding these rare species is really helpful if you have field experience, and it will increase as sampling goes up. So Dean collected 109 rare species total, and I collected about 17 rare species total. Given Deem's number, again, extrapolating here roughly, I should have collected around 45 rare species. But again, I was collecting a lot of common species, trying to get them out of the way for the next 2 field seasons, so there's a bias there. And it'll be really interesting to see how these numbers change in the next 2 years. So moving on to the county records, overall I found 152 of them. These are just some examples. So here we have Viola striata from Posey County. We have Clematis virginiana from Tippecanoe County. And we have Phacelia pershii from Knox County. And we do not have enough data yet to statistically address whether native species are moving latitudinally in correspondence with climate change. But we do have some observational results. So these are both state listed plants that occur in the southern part of the state that like disturbed habitats, and were found in only a couple of locations when Deem did his survey of the Indiana vascular plant flora, but now have seemed to move northwards. And I documented them in counties in the southern part of the state where Deem did not find them. So to summarize the results from the first year, there are many exotic species, more invasive species, and not quite as many rare species. And to address the more sophisticated hypotheses and to provide a more statistical approach to these 2 previous questions, I need to greatly expand the collection scope. So I need to collect probably anywhere between 15,000 and 20,000 vouchers. And right now, I'm currently in the process of trying to get funding to do that. So this is something that is in the works. These data points, as I just said, will enable a much more comprehensive statistical analysis for these more complicated research questions. And I couldn't get through this talk without including a little cicada. So I have him there with a plant that turned into a specimen up in Tippecanoe County. So now I'll move on to one of the more interesting parts of the talk. This is the future directions. This is addressing more sophisticated hypotheses once I have the full 3-year collection data. So the first question that I'm really interested in is, are native species moving latitudinally in correspondence with climate change? And one of the things I've already been doing to address this is I've been working with putting together a statistical approach to address this question using existing methods and statistics. We also need more data to answer land fragmentation and quantitative biodiversity questions. So with climate change, as I said, I'm working on this novel approach that puts together these existing statistical methods to address this statistically. And to do this, I'm using probability models and exact multinomial tests. And the output from this process is statistically supported climate-based floras. And once you have a statistically supported climate-based flora for a specific time point, you can compare the climate-based floras at a different time point. So to illustrate what a probability model is, I have the state of Indiana here, and this is mean minimum January temperature for the Deem collection period. This is the same thing we saw earlier for the study period, for the study region, except now we have the northern segment as well. So we have 6 1°C minimum intervals here. And I've weighted the probability that a plant occurrence happens in any one of them by the number of counties and the land area in each county. So if this is the A segment, the probability that a plant occurrence takes place there randomly is about 21%. And so you can go on down the list. And with plant occurrence data, we can see using an exact multinomial test whether this expectation of these percentages is met or if it's significantly different than expectation. So here's Gallium pilosum. It's a species that's significantly different from this expectation that observations should be equally distributed among the climate segments because it has this very large break in observations in the middle of the state. Now, historically, it's likely the species was contiguous throughout the state, and because of major deforestation in the middle region, has since been extirpated. Now, we can go a step farther, and we can take these species that do not fit the null model, that they equally are distributed among the climate segments, and we can test them against alternative models. So we can classify them to specific climate segments. This is where we get climate-based, statistically supported floras. So here in the lower right-hand corner, I have the climate segments for reference. And then here I have examples of models that fit alternative models. So here's a buckeye species that occurs only in this next to the most southernmost segment in the state, the Eve segment. I have a dodder vine that occurs mainly in the two southern segments of the state and it has some seemingly random occurrences in the northern part. And then I have a skullcap that, despite only not occurring in a narrow swath of the northernmost part of the state, this approach still flags it as significant and classifies it to a different alternative model. This is a very powerful approach, and we can see if these distributions change over time using this approach, by setting the historical distribution as a null model. And it's important that we also have variation in minimum temperature over time in order to say, okay, well, maybe it's correlated with temperature. And here is the mean minimum January temperature for a more recent time period, 2010 through 2020 or 2020. So here we have only 5 1°C intervals in comparison to the previous 6, and we lose this -10 to -9°C interval. So now we're able to see if species that met this this model, for example, have moved northwards in correlation with the change in temperature by seeing if they fit the same alternative model for temperature that they fit historically. Now, this may be very confusing. This information really should be its own talk, but if you have any questions, I'd love to talk about it afterwards. Moving on to land fragmentation, we can address the question above by generating a site by species matrix here, and we can incorporate site data, so whatever variable we're interested in. So if it's land area, we'll include acreage for each site, and then we can construct a general linear model that we can use linear regression on to see if the variable is correlated with diversity. And here I have the general linear model or linear regression equation. And we can do this for nonlinearity as well. So for nonlinear relationships too. Moving on to beta diversity. So before I mentioned this, but I didn't explain what beta diversity is. So beta diversity is simply a ratio between regional and local species diversity. So it quantifies the difference in species composition between sites. So once we know how sites are different in species composition, we can test different variables to see if they correlate with these differences, to see if they explain these differences. So examples are down here. So we could test for soil type, ecoregion, climate, etc. And we can use regression as before. So here's a visualization of what beta diversity looks like in woody tree species in Indiana. And this is from Deems data, and this visualization technique is actually called Ward's clustering, or that's actually the beta diversity metric Ward's clustering. And here we have essentially the northern counties all break into one group. The center counties break into one group, and the southern counties break into one group. And it's important to note that the center and southern counties have more similar woody tree species diversity than the northern counties do to the either one. So it's interesting. It's important to note that you can bootstrap values that provide confidence values for each of these nodes or branches. And for all of these minute branches, they're not significant. But if we move up the tree, here, the northern, center, southern branches are all statistically significant. So there really is this relationship between the center and southern, and then between the two to the northern beta diversity in woody tree species here in Indiana, at least according to the Deems data. So moving on to phylogenetic diversity. Phylogenetic diversity is simply a measure of evolutionary history represented within a community. And some biologists argue it is more important than simple species richness because it better reflects functional trait diversity. But it is more difficult to calculate because it requires sequence data, and it's also usually inferred because it's highly correlated with species richness. So I also did some data analysis for a course last spring with another graduate student looking at woody tree species phylogenetic diversity across the state. So here on the left, we have just the number of woody tree species per county plotted. And then here on the right, we have the FAITH's phylogenetic diversity metric for phylogenetic diversity. And you can see that in both the southern parts of the state and the northern part of the state, both metrics are high. But the middle part of the state does not scale evenly. So some of these counties are more phylogenetically diverse than we would have expected based on just the species richness. And that's because across the phylogenetic tree for woody tree species, there's good representation in these counties, or better representation expected probably by chance. So I want to go ahead and conclude my talk, but I want you to take away that this floristic analysis of the Wabash River corridor is not just relegated to simple questions, but with the combination of statistics, we can really address more sophisticated hypotheses to determine how the Wabash River corridor has changed since the early 1900s. This will allow us to identify general trends that may apply to other systems, whether it be geographic trends or taxa trends. And the information garnered from this research will enable me to make more accurate predictions on how the flora in this region will change in the future. This information, as well as the status of exotic and rare species, will be made available to the Indiana Department of Natural Resources, who have a practical impact on conservation here in the state. So I would like to thank the Indiana Department of Natural Resources for their cooperation, and as well as landowners for all my sites. I'd like to thank my PI, Dr. Eric Knox, and Dr. Paul Rothrock for helping me with identifications, and several of the people who've helped with working on the voucher specimens, whether it be mounting or identification, and then working with my photographs as well. And lastly, I'd like to thank my wife, Sophie Hull, for listening to me rant about plants pretty much every day. I'd like to thank my funding sources, the Indiana Academy of Science, and then the Department of Biology here at Indiana University. Here are my references, and then I would love to take any questions if you have any. Thank you for listening today.
Rich, thank you so much for sharing this information about your research and the current and future stages, and also giving us some insight into the IU Herbarium and the larger consortia of which it's a part. I took a moment to poke around in there, and it really is a fantastic resource. So I am going to take a moment of sort of host presenter's privilege here and ask you a first question while we are allowing other folks to have a chance to pop anything that they want to say or ask into the Q&A. And I will just say that I'm a humanities scholar, and a lot of my work is collections based as well, but a slightly different type of collection. So it's coming from sort of more of a lay person outside the discipline perspective. But that's just to say that one of the things that I love about collections and research that intersects with collections, I think is really important, is that it allows us to have ongoing engagements with researchers, scientists, other scholars of the past, and to bring that to our work as well. And so I wondered, one of the things that I know from talking with other colleagues about previous projects is that one of the things we can learn from collections is not only the data about what was there at a particular time or what work was being done, but we see difference in methodologies as well, and the types of inquiry that are happening. And I wonder if you just have any little anecdotes that you want to share about that, things you're seeing when you're looking at Dean's data and, you know, your data and that kind of stuff.
Sure. So Dean's data is actually very different from the data I'm collecting. So his goal was to generate a complete species list for each county, whereas mine is a complete species list for each site. And in data analysis, obviously that presents certain challenges. But in statistics, usually we can weight the number of sites per county or a number of sites per latitudinal segment to account for that. But other than that, I mean, there's really no direct conflict there. I mean, we can account for it statistically. And the reason why I'm diverging from Dean's county site level analysis is that using specific sites enables us to address questions regarding habitat fragmentation and beta diversity between sites, rather than just thinking about, let's say, a county as a site. It's not very helpful.
That's great. And I think you also mentioned in your description some of the other things that can happen, which is when there are actual specimens as opposed to just photographs or illustrations in previous times, then later people can come back and learn more about that, or even say, wait a minute, I'm not sure that this initial identification is accurate. There's this other feature here. Or I think you mentioned that some of the specimens have been renamed in the years since they were collected. And so that those types of things, I think, also kind of highlight the real importance of having these collections and resources available. So thank you for that. I'm going to switch over now to the questions that are coming in here. So the first one says, if you collected roughly 2,500 vouchers in one growing season, what's the plan to collect 5 to 6 times of that amount over the next 2 years?
Well, the plan, number one, is more funding. And that funding will go towards hiring another field technician for this next year and hopefully the year after that, which should at least double my production. And generally, I also have more logistical modifications for how I'm collecting. So instead of, let's say, collecting for 2 days on the road from Bloomington, in order to alleviate travel time, I'll probably collect for 4 days on the road from Bloomington or something along those lines, which should also have a large effect on the number of collections. The last thing too is that the goal is to generate a full species list for each site. That's obviously not going to happen, but there's something in ecology called a species area curve, where you can look at the number of species documented, you can look at the number of the area of the site, and you can predict the number of species that have not yet been documented. So that's something we'll use eventually as well.
Right, and I think in any project, that first stage of the data gathering is the one that takes time, as you're refining the methods and figuring out exactly what's happening there on the ground. So good luck with that. And we're definitely hoping for the funding to come through for you as well. The next question is, how will your work on the Wabash River corridor apply to other counties in Indiana and the broader Midwest?
So this is obviously a small section of Indiana, but generally we're going to be able to say, well, are species moving, in accordance to climate change, and maybe what are these species traits? Are they species that are well adapted to disturbed systems? And if so, we expect the same thing throughout Indiana and the Midwest, so similar trends. Oftentimes in climate change research, these traits are relegated to taxa within a group. So let's say a genus. So if we find one genus is doing one specific thing, we can predict that genus is probably doing the same thing in the broader area. Otherwise, the impacts, let's say, like if we think about land fragmentation, if we find that the time since last disturbance is more important than the land fragment size, that's probably going to be broadly applicable to systems that are nearby as well. So these quantitative biodiversity approaches will also enable us to see how plants in the region are reacting to human disturbance.
Excellent. So I'm going to take another opportunity to go back to one of my questions while we see if there are other folks who want to chime in, which is earlier in this semester in this talk series, which you're now participating in, we had a presentation by David Polly, who's with the IU Paleontology collection. And so he was talking about what to me feels like a similar kind of sampling in the sense of they were looking at animals and taking surveys of animals and the number and variety of animals, and then comparing those with the data that they have from past times. So my question is, are there opportunities either here at IU or more broadly out in other scientific communities where those of you who are working in botany and those who are working in paleontology or zoology or related disciplines are able to compare those? Is it useful to compare those types of surveys and data, because they're also taking into account climate change and species change over time?
Sure. I mean, I think it's really important to think about. I'm not aware of a specific example of this. It's actually very rare to see, I would say, to see collaboration between like a botanist and a zoologist looking at historical data. But it'd be interesting. It would be a cool thing to consider for me going forward if this is happening in Indiana.
Yeah, sure. And we'll make sure that you get to see the link to his talk and he'll get to see the link to your talk, and we'll share the information that way. One of the things we like to do at the Institute for Advanced Study is bring together people who maybe have overlapping interests. So we have another question comment. It says, excellent talk, Rich. I have to agree. It says, I'm curious to know if plant diversity is related to insect diversity, for example. Thanks.
Oh, certainly they are, yes. Because plants are primary producers in many terrestrial ecosystems, and because plants are so closely linked to insect diversity, they definitely are. How I can't really quantify that per se, but there are many specialist insect species, which basically means they require one plant for their larvae to feed on at a stage of development or something along those lines. And you just won't have that insect species without the host plant species. So tree diversity and plant diversity broadly is extremely important for maintaining insect diversity.
Thank you. Our next question says, Indiana is relatively flat. Would responses to climate change be different in mountainous regions?
Yes, they would. One of the main reasons why I'm doing this research in Indiana that I did not mention was actually that Indiana is relatively flat. So most climate change research on plants has actually happened in mountainous regions. And this is because changes in elevation lead to greater climate changes more rapidly. So plants are able to migrate shorter distances in order to achieve more effect essentially in climate change. So it's easier to determine when a plant moves, and it's more likely a plant will move over a short distance. But the majority of terrain on Earth is actually not mountainous. So despite it being difficult to pick up on these changes statistically, and it requiring a much larger data set to do so, I think it's very important that we do it. And that's really one of the other main reasons why I'm addressing this here.
So then I'll just ask a follow-up question to that, which is the difference in terrain between northern Indiana, which we know is particularly flat, and southern Indiana, which is frequently referred to as very hilly rolling, is that enough of a difference that you see a real difference there, or it's not dramatic enough for that to have the impact that you were just discussing?
It's not what I would think of as mountainous. It wouldn't really probably have those effects. So where people are studying traditionally mountainous terrains, like the Rockies or some analogous mountain region where we have this like decisive ecotones where you can see like the vegetation decisively change at different elevations. You get some of that in the Appalachians as well. But in those cases, you have to account for plants moving both latitudinally and in elevation. So it becomes really complicated. But I don't think in Indiana generally we have to worry about elevation all that much. But if we think in terms of just climate, and if we have finely gridded data, we can address it.
And somebody is curious if you have ever run into a bear in the field.
Yes, once I did. I was with my dad and we were in northern Georgia and we were in this very steep ravine and he decided to go up the ravine and I decided to walk in a creek at the bottom of the ravine to try and find a better place to go up, and he flushed a bear down towards me. It was just rumbling down this ravine and yeah, I kept running. Yeah.
So which of you likes to tell that story more often?
I probably do.
Yeah. Someone else says, is the widespread land conversion to agriculture in northern Indiana an impediment to your research questions?
I don't think so. It's actually one of the reasons, like if you think about the human activity here in Indiana, as one of the reasons for doing this analysis, because it will highlight the degree of resilience native flora has to these activities. Obviously, if you can just convert everything to agriculture, it will wipe out the native flora. But in terms of land fragmentation questions and quantitative biodiversity questions, it's helpful. With climate change questions, it could very well inhibit the species' ability to move latitudinally. But at the same time, if we look at this from that perspective in a system like that, if we still see species moving, it means that they can move across habitats or landscapes that are highly fragmented, which is helpful information. And it would, if that is indeed the case, which is probably somewhat unlikely, it would be very good news.
That's great. So I'm just looking to see if we have any more questions or comments. Give folks just another moment. I'll take this opportunity to note that you mentioned Paul Rothrock, and we will have a presentation coming up next semester, I believe in January, and Paul's going to present about the photo scavenger hunt that he and Eric Knox worked on with others, and that was part of our repository research program, our larger program. And so that involves citizen scientists from around the state taking photos of species to be added to the database. And we're really looking forward to that. So we would encourage anyone who's interested in these types of topics to check out our website at the link that was mentioned above. Keep following that so that when that link is available, you're able to register for that webinar as well. So we are really looking forward to that too. And I will just take this opportunity again to thank you, Rich, for your time, sharing your expertise with us, answering all of our questions. We are really glad to have you with us as part of this talk series. And also I want to say thank you to IU Semester, who has supported our Resilience in Memory in Archives, Libraries, and Museums series this semester. We appreciate getting to be part of the themester for that. And we are looking forward to continuing with more conversations about repository research in a variety of fields in the future. So I'm not seeing any more questions or comments right now. So I think we will say thank you again to our presenter. Thank you, Rich. Thank you to all of you who have joined us. We hope that you will join us in the future. If you're interested in finding out more about upcoming IAS events, please email us at ias@indiana.edu. We would be very happy to add you to our email list so that you receive notifications of these things. You can also follow us on Facebook and find us at our website. So thank you again for being here. We look forward, Rich, to seeing how this project develops. Thank you again.
Thank you.
Take care, everyone.